introduce one of our own, Xiangmin Xu, right here for today's Center for Virus Research seminar. Just to give you a little background on Xiangmin, he got his undergraduate and master's degrees in Shanghai, and then he came to the US to Vanderbilt University, where he received his PhD in 2004. He studied the functional organization of the primate visual cortex during his dissertation research. And then he came west to the Salk Institute to carry out his postdoctoral research in Edward Callaway's lab. And in Callaway's lab, he worked on the properties of different classes of inhibitory neurons in mouse brain. In 2008, Xiangmin joined the faculty at UCI as an assistant professor of anatomy and neurobiology, and he's now moved up through the ranks to the rank of full professor. He holds joint appointments in microbiology and molecular genetics, in biomedical engineering, and in computer science. And most recently, he was named director of a new School of Medicine, UCI Center for Neural Circuit Mapping, which promises to further enhance the already stellar neuroscience research activities here at UCI. His research at UCI is focused on a range of important neuroscience topics broadly related to understanding neural circuitry, including plasticity in the visual cortex and cell type connections to the hippocampus. Of course, he has to work on the hippocampus. He's at UC Irvine. So as you will see in today's seminar, he is developing and implementing state-of-the-art technologies to engineer recombinant viruses that allow tracing of neuronal circuits. During his academic career, Xiangmin has been a recipient of a number of awards, including the Narsad Young Investigator Award from the Brain and Behavior Research Foundation, the Randolph Blake Early Career Award from Vanderbilt University, the Creed Cortical Explorer Award from the Cahal Club Foundation, and the C.J. Herrick Award in Neuroanatomy from the American Association of Anatomists. And very recently, he was named Chancellor's Fellow here at UCI. He is very active in peer review of manuscripts and grants, and was recently appointed as a standing member of the NIH Pathophysiological Basis of Mental Disorders and Addiction Study Section, or PMDA. He's been an incredibly productive researcher during his 12 years at UCI, and he has a truly stellar record of peer-reviewed publications and NIH grant fundings. So today he will give us some of the highlights of his research program in a seminar entitled Genetically Engineered Viral Vectors for Neuroscience Research. Xiangmin, take it away. You're muted.
Well, thank you, Dr. Sommer, for your very kind introduction. Yes, I will treat your words as my encouragement going forward. Hello, everyone. And I'm very pleased to have this opportunity to give a talk at the Center for Virus Research Seminar Series. So as you heard in Bert's introduction, I'm not a virologist by training. So the purpose of today's talk is not so much about the viruses themselves. Rather, as a neuroscience research, I will present why we use or why we need viral tools for neuroscience studies and what we are doing to develop new viral genetic tools for neuroscience studies. And then a second point is that I do consider these great opportunities. I consider these opportunities for to expand my interactions, collaborations, particularly with viral experts at the UCI community. Okay, let me go ahead to share my screen.
Right. Looks good.
Good, thank you. My talk title today is Genetically Engineered Viral Vectors for Neuroscience Research. Before I get to this topic, I would like to provide an overview of my lab research programs. So my lab interests are centered in the neurocircuitry. And we are interested in the neurobiology of sensory perception, particularly visual perceptions, learning and memory. Understanding how neural circuits give rise to perception, cognition, and behavior is essential to understanding how the brain works. And this is also key to understand, to achieve the mechanistic understanding, the basis, substrate basis of neurological and neuropsychiatric disorders. So my lab, we have a broader range of funded research projects. And we study visual cortex, prefrontal cortex, we study cortical structure and persistence. And also we have a significant number of projects to study the hippocampal neuroscience, including aging and Alzheimer's disease research. And as Bert mentioned, that I have multiple joint appointments, and this actually is pretty important because the lab really taking the multidisciplinary approaches. As most relevant to today's talk, we are very strong in developing new viral genetic tools for neural circuits mapping. And our research, I do think our research has significant potential for translation applications. In terms of our the visual cortical studies, this is like research. So we believe, really has potential to apply these findings to better treat the childhood visual disorders such as amblyopia. In lay terms, amblyopia called lazy eye. And on the other hand, the hippocampal studies, I think this is something I'm very excited about. We hopefully will be able to apply our new circuit study finding to better treat the Alzheimer's disease. And we are substantial. Currently we have substantial NIH funding to support this line research. So with this overview, I'm going to get into today's topic. So, first I will introduce you a little bit background, neuroscience background, and tell you about a brief history of brain mapping and the history of the virus being used for neurocircuit studies. And then I will describe some recent progress in engineering the viral vectors in contact with neuroscience research, and particularly I will illustrate how we use these new viral genetic tools in circuits application, circuit mapping applications. And finally, I will describe our current efforts at the UCI toward developing new viral tools, moving the field forward. So brain mapping is a critical area of neuroscience. So origin of the modern neuroscience can be traced back to late 1800s and early 1900s. As shown up in this slide here, one landmark work was done by German anatomist Korbinian Bodemann. So the bottom here, this shown here, this basically is well known. The Bodemann's work of localization of the different areas in cerebral cortex. But these Bodemann areas, this is also the Bodemann basically define those define those areas, give numbers. So they define and number these areas based upon histologic characteristic, or you call it, cytoarchitectural. In this case, some people call it cytoatonic, basically cytoarchitectural differences. So they're based upon the cell size, organizations, to divide the cerebral cortex into different sub areas. And this amazingly, interesting thing that even those are structurally those divide sub areas, those brain areas in terms of histologic characteristics. It later on be turned out to find a piece very much highly correlated with function. Right? For example, areas 1, 2, 3 correspond to the primary sensory cortex in human, and area 4 essentially here is corresponding to primary motor cortex, and the area 17 correlates to one of my favorite areas, that's primary visual cortex. And so these, so those real landmarks, is a layer basic foundations for modern neuroscience. However, those maps really tell you the map, does not really tell you the connections, other things, right? And there's so the second major breakthrough event, I think, really important is the Golgi staining, right? So Camille Golgi was an Italian histology and pathologist. And he accidentally just invent or discover this staining technology. And the Golgi technology is kind of very interesting, So Golgi technology is just, we still don't know the exact mechanism, how these kind of reduced silver staining would be able to just stain a subset of neurons in entirety, including those elaborate details, such as you see those, you can search some of those dendritic spines along the dendrites. And so these, the Golgi methods is really, I think it's revolutionized the field, because these first time allow the scientists or investigator to see that, to visualize neurons, the fundamental unit of our nervous system. And so then, because this staining or subset, it will allow the Golgi to draw the first map organization, basically first map or anatomic organization of hippocampus. And more than that, the Golgi staining methods will allow the earliest surface analysis of neural structures by Santiago Ramon Cajal. Cajal, because of his fundamental contribution to neuroscience, he was considered as the father of modern neuroscience, and he shares the Nobel Prize with Golgi in 1906. Cajal's brilliant, right? Cajal basically used improve and applies the Golgi methods, and to basically, he draw a lot of things, he visualized the circuit structures. Right? Even we don't understand that time, even we don't know if neurons, or how neurons organize, and there's a, Golgi is totally different dominant view. And Cajal proposed the neural doctrine based upon his careful observation of the Golgi staining of neural elements, neural circuit connectivities. And as illustrated here, this beautiful drawing of the hippocampus, right? And so you can see those are those neural fiber getting into the area of the hippocampus called dentate gyrus, and you have arrows. It means so it's really, because at that time we don't know nothing about the the brain system, not alone the hippocampus, and he basically kind of, but it's basically imaginary, right? It's the arrows say, well, these are how the signal transport forward, and turned out all the fundamental principles Cajal discovered at that time was later on being proved to be true a few decades later. And still, so so as I mentioned, it's very amazing. Cajal's use, his essentially his imaginations to figure out, to lay out the circuits connections. And so really, there's very limited experimental technology to do tracing the brain, even in terms of the inter-brain or cross-brain region connections. And only one, the earlier between somewhere around, before the 1950s, is really people been using called, essentially is integrated axonal degeneration, volume degenerations. I use these to tracing axonal pathways. So I will take this opportunity to introduce about some basic elements of the neuron. So neurons is a specialized cell, right? It's with certain priorities. So cell body, called soma, and dendrites. Usually we believe that dendrites is, so the neurons receive their input. And the neurons receive input, and then they have, if it generates action potentials travel down to axons, projecting, basically projecting the output, right? So this is basic output, the send signal out. See, this output and this input, output. And there was, that time was known that if you cut axons, those axons somehow will degenerate, and the important thing that those degenerative axons, they have a much higher affinity to the silver stain. And now there's really one the key person to develop the methodology can suppress the cell impact in normal axons, but basically enhance the staining for the degenerate axons. That's provided the contrasts between healthy and degenerating neural circuits. So, with a little bit of practice to trial and other things in conjunction with the brain region, with good timing. Timing is important because also degeneration can be eventually will be absorbed, cleared by the glial cells. Right? And so we use this axon axonomy and the civil staining, good timings. Early neuroscientists kind of map out a certain cross long range connections between different brain regions. Right. So these are so anyhow, so you can see the history is basically gradually just making small progress, right? And so the one of the golden areas in modern neuroscience is starting like late 1970s. Right? That had more effective methods, particularly chemical tracers, invented for tracing the axonal pathways, right? And those including the angiograde. So the angiograde basically radioisotopic and amino acid treated amino acid, and further on later on they developed the HRP, so horseradish pyruvate HRP, and some of the angiograde, including the PHAL. So those are radioactive amino acid PHAL we call the angiograde tracers because they get into the cell body, right, of the neuron and gradually transport, along the axons, right? So this is what we call the angiograde tracing, because angiograde go into the axon, and you can do the staining, or you can do audio radiography. You can be able to see the axon combination sites. So based upon these things, people can figure out where this brain, brain region part, or where this brain region part to other parts of the brain. In comparisons, and so that people also have those traces called retrograde traces, including HRP or adductions. And so retrograde means that the axon terminals uptake those traces and transport back to cell body. Right? So you can see here, so can nicely to label the cell body. Right? So by using both much more effective methods of the retrograde and antigrade traces, and neuroscience actually make great, very impressive progress in the last 4 decades also. And those chemical traces are essential for developing our current understanding of the neural anatomy and the global neural connectivity wiring diagrams in the brains. So I took these illustrations from Larry Swasson's work. So Larry Swasson's he's a retired professor right now at USC. So he done some of the pioneering substantial work, a lot of the connectomes, particularly in rats. And with all these understandings, so even these days, even around 2005, people come up with a term called connectome, right? And so now these become very popular terms. And so you can look at the connectome, essentially look at the matrix connections between different bone regions. Right? So everything's good. Things looks pretty good, right? So however, those maps is still pretty much overscale. Still give us it's macro wiring programs, wiring diagrams, and those traces still very much it's good. We're not those traces are significant limitations that they don't have significant sufficient resolution to understand cell type specific. So basically, we're not allowed to see the connectivities at the level of molecular defined cell type. Or they don't have, also they don't know because you usually just uptake, but you really don't know how these the map cells connect with other cell type, but within the local bone region. Yeah. So this really is the motivation for us to engineer viral vectors for neuroscience research for brain mapping. Because compared to the non-viral chemical traces, virus can be used to target specific cell type, use genetic strategies. We can use specific expression of viral receptor, essentially is pseudotyping, or we can express recombinant protein specific cell types to direct, the productive viral infections or payload delivered to certain cells. So those technology enhancements in the last 2 decades also really start to start another wave of revolution, and it's also increasing viral genetic tools that will replace the chemical traces. And it turned out it's very interesting, right? And so those use of the viruses, for neurocircuitry tracing has a long history. And a lot of works actually were done by virologists. And for example, so the Ernest Good Pasteur was a pioneer in the field of virologist. And really the work I think is really starting in the 1920s. And Good Pasteur, his teams will provide this earliest support for herpes transmissions to the brain. So it's really through the optical nerves to produce the experimental encephalitis in rabbits. And in the 1930s, Albert Sabin, who later gained the fame for the development of the live attenuated polio vaccine, made this important observation that neurotropical viruses, in this case is herpesvirus, enter the brain through differential neural pathways in mice. And these are very important observations. It was followed by the earlier neuroscientists in 1970s and 1990s by demonstrating the axonal transport of the herpes virus to neural cell bodies, the transneural spread in the central nervous system. Okay. And some of the earlier studies really highlight a great advantages of using viruses. But even use the even use those genetically modified viruses. And because of the virus, also we know that virus is great too, because the virus can replicate themselves within infected neurons. So this really allow, so it's not like the chemical traces, once you uptake, you will not enhance, basically once you take it, your traces become dilution, right? So normally it's only allow you to do, uptake. You only look at the visual uptake, even most time you need to enhance to staining to enhance them, right? Well, virus is different, right? Can self-replicate. It's basically where virus, we can use virus as self-amplifying markers. As shown, those illustrations are taken from the earlier work by Hans Kuppermann and Gabriel Ugolinius. Particularly Ugolinius had done a lot of work, pioneering work in use virus in animal research. So let me explain it a little bit here. So these basically showing here that they inject the herpes simplex virus type 1, HSV1 here, into the hypoglossal nerves. And these viruses were uptaken by motor neuron, retrograde, in this case, retrograde. And then, so you wait for a certain time, and then the virus propagate and go into the, theoretically, we're getting into the connector neurons, further going back to retrograde. And I really want to point out here, these also are those are labeled neurons. So what do they, because these are wild type of virus, how do you visualize it? You have to basically, in this case, you have to stain them, to see. And so these, you can see those dark spots are those are viral infected, lots of the viral proteins, and then you also look at this second order, first order, this injection site, and this is the labeled neurons. You can see that they actually don't dilute. This is very prominent, highly prominent. And also those neurons is actually, it's not specific, it's projection specific. Those neurons are uptake, basically they're porting to these injection sites. And so this, another thing that once you stain them, they're pretty impressive. So they're kind of comparable to certain, to the Golgi stain in certain cases. So this is basically, those kind of early work really generated enthusiasm, right? Because now we have some new traces. They can self-replicate, right? And can even in this case, it can go through dependent time. It can go to one order, second order, even third order. Right? However, the earlier virus, those experiments also suffered a significant drawback. Right? And one thing is that those wild type of virus, we don't know how they spread. Do they really go into the retrograde disease way, or they may spread it different ways? And also the virus high toxicity. If you look at injection site, they kill a lot of cells. May potentially those viruses will spread, lateral spread. You lose the specificity of the synapse spread specificity. Right? Those are major drawbacks. And in addition to the using herpes virus, and usually the German group as well as the group in United States pediatrics and also applies rabies virus in rodents, in monkeys, pediatrics, and a lot of work in monkeys tracing use rabies virus. As is here, anyhow, very similar. You inject it in the muscle or nervous, you look at labels, first order neurons, and gradually all the things spread based on the time, right? Based on the experimental evidence. So how long wait cause uptake of one synapse to second synapse. Right? And so if you look at it here, if you do see rabies actually is better, much better traces. Somehow within a few days, they actually don't cause, very apparent cytotoxicity as compared to the herpes. But anyhow, so the earlier the virus work for neurosurgery tracing generating enthusiasm, but there are people kind of and a lot of people still neuroscience still remain highly anyhow people still are not very convincing or these would be good thing to use virus for tracing until the late 1990s. This is really a turning point. And turn out attenuate the wild type. This is the wild type of the pseudo-rabies virus called Bassa strains. Although it's attenuated, it may take a little more time to label those neurons, but it was shown that we produce highly specific retrograde transneuronal infection. And turned out the reason why these Bassa strain would be highly specific retrograde because there's the natural genetic mutations, right? And it's one real mutation really is called US genes. These basically block the antegrade spread of PRV in neurosurgeons. And a lot of works done, this is basically works done by Lynn Anquist's group at Princeton. And so the PRV, the PRV Bassa strain is really in critical turning points for renewed interest in seriously using the virus, genetic modified virus for such tracing. And then subsequently, the PRV being further modified, particularly added, the different type of fluorescent labeling. This is really important because the virus, the staining is just, it's just not a good way. And you have highly sensitive fluorescent proteins that work much better. And the PRV has, I just mentioned, the virus is very interesting, for the pseudo-rabies, they affect rodents pretty well, but they don't affect the non-human, don't affect the primate species, including humans. And the those genetic modifications lead to a large improvement, not just pseudo-rabies, and also the rabies virus, herpes, and herpes simplex virus. And in addition, the marker gene, and also control. There's in terms of how many synapses usually polysynapses. It's for a lot of experiments that don't work very well. People would like to look at it as monosynapse spread and other things. And also the pseudotype is very important. Allow certain selections infection of certain cell types. And so mostly what I talked so far is, you can, if you want interesting more detail, you can refer to our recent published primary article in New York. I would like to really want to highlight to say this is a group, is really the group team efforts, right? So including number of the virologists, including our Dr. Sanjay Golden, Dr. Boda Semra, and we have a Chinese colleague, Ming Fang Ro, and Ming Fang's staff, and as well as my long-standing collaborating colleague, Todd Holmes. Let's see. And so in the next part, and so I'm going to tell you the recent progress and the new circuits mapping applications using the genetically modified viral vectors. So currently, the field is used mostly, so people use a lot of different viruses, but the genetically modified AAVs, lentivirs, adenovirus, and particularly the neurotropical virus, herpes virus, rabies virus, primarily the vectors are using for studies. AAV, including AAV, lenti, adenovirus, and most obviously these vectors belong to the category of the vectors for gene deliver and cellular label. And this is important, cellular label, because it also allows to label that they can fill the particular marker gene or fluorescent protein can fill the whole cell body, including exon there. So people can use it to do the external tracing, external projection tracing. And so AAV, I think most people are familiar with AAV. AAV is a single strand positive DNA. The genomes, where the capacity is relatively low, it's only 5 kb. But anyhow, they are easy to package. And another thing is, for neuroscience, AAV is nice because they really don't have much toxicity, lack of pathogenicity in animal models, and have relatively long-term expressions. I mean, they have relatively broad tropisms. Right? So those are bigger. And it's DNA virus. That's very good because we can use the Cre-LoxP system, right, or the basic other type of the recombinases to do. Consider we have a lot of the Cre line, Philippus line, really easy to use AAV for cell type specific targeting. And so AAV, so I just want to show a couple of examples, see how people have been using AAV in recent years for connecting mapping. And these illustrations taken from the Allen Brain Institute the publication Hong Kui Zhang's group from their 2014 Nature paper. So in this case, what they did, they used the fluorescent expressing AAVs, either Cre version or non-Cre versions. In this case, so they inject one mouse, inject one bone region. And then they can section all the brains, image them, and put them into a standardized brain template. So and it smooths out. You can know if you inject this as orbital cortex where there's projection. So also really look at the axon, right? So brings the EJP express axon labeling. And similarly, they inject different mouse, inject different area, and you can create a kind of brain map, bone map. And so this large scale project, they inject hundreds of different regions, 100 different regions. And it's noticed as beautiful, some of the beautiful axon projections. And so so once you processing all the data, put them all into the standardized brains, and now you have a much higher resolution of the interbrain connectivity map. So this is one of the great example to show the progress using the new genetic recombinant virus. And more recently, the mouse light project it's in the generic form. And so that project, so it's here to be shown that the project reconstructing about 1,000 neurons, those project neurons. And the nice is different from Aaron's that they actually use a sparse label. In this case, so this sparse label, a couple of those significant neurons in one projection, in one region, in this case, the significant. And then they do brain clearing, there'll be imaging, This unprecedented, those, it's really the resolution of the axon projection is unprecedented. And so, and then they do I put all the tens or hundreds of cells into a standard brain map. It allows us to really see the different, they argue they would identify new cell type and also organizations, long range connectivity in the mouse brain. Right? Those are large scale projects. It doesn't require several years teamwork. It's expensive, but the data is generate totally new insights in terms of the specific cell type and the brain, the inter-brain long-range connectivity. However, as I told you, the AAV is good. The vector is good for cellular labeling, for gene payload. But those really tell you the axon projections. I really don't know what cell type, what's the postsynaptic. They do not really they're not vectors, transneuron or transsynapse. So all this work will need to be further done, further improved using a different type of vectors, the transsynaptic neurons. Right? So I actually will skip it anyhow. So just again, there's two types of the transsynaptic viral traces. One is retrograde. You start from a cell body, you go back. Retrograde from cell, so one through the synapses and go into axon, go into cell body. It could be monosynapse or polysynapse. And similarly here, this is called antegrade. Antegrade, you start with neuron here, axon project to dendrites, and there's so synapse go second, first order, and potentially go to second order, depending on the viral traces. So yes, the AAV work well. It's produced very nice data. But next level, I understand we really likely going to look at further to use the new transsynaptic tools to use this. And so really, we have relatively limited number of the virus can do so. But the most basically has the herpes virus, the herpes virus, including the HSV1. Advantage is that they have large capacity. The vector capacity can up to 50 kb. That's you can do a lot of things. And they're neurotropical viruses, so showing the strong neurotropism. And there's multiple replication defective or replication competent strains. For us, very much interested one that HSV have one called special strain called H129. And these wild type H1 strain, they show antegrade preferential propagation. So this is something we've been actively working, trying to genetically improve to develop the antegrade monosynaptic traces. So the significant disadvantage is that the herpes or the general herpes, including the PIV pseudovirus, they're highly toxic. Right? They not just kill cells; they kill animals in a few days. Right? And another thing, there's a spread. There's concern still. There's a huge concern about use of the herpes virus is about their specificity of the direction projections. Right? It's a retrograde. They could potentially have both retrograde and antegrade. That's really limited the interpretation of the tracing data. We already discussed about PIV, but it's actually pretty good for anatomical. But anyhow, the toxicity is still limited them for study the physiology or function studies. And there's the PIVs that don't infect the primates. So the other major category of virus, transsynaptic virus, is rabies. So rabies is a specific retrograde transsynaptic traces. Particularly the genetically modified rabies-based monosynaptic tracing system has been well developed and widely used. And it's rabies. It's interesting. Rabies. relatively high toxicity, but they're okay for short-term studies, right? Because they are so in the study published, so you can do January, you can do anatomy within 2 weeks. So okay, even you can do electrophysiology about 7 days. Right? So their toxicity, they are not as bad as the herbivirus, but still for the longitudinal studies, we need to further reduce the rabies toxicities. These are some things. Is some of our current focus I'm going to talk about in my final third part of my talk. So Dr. Edward Caraway is a pioneer in developing and applying genetically modified monosynaptic rabies transient system for neurocircuit study. So Ed, because Ed actually do a lot of things, but I think the rabies tracing is probably one of the major factors for him to be elected to the National Academy Sciences in 2019. And as Bert introduced me, so I was training with Ed between 2004 and 2008. And the Salk Institute is a great place, for postdoc training. And Ed is very kind. We learn a lot of things. That's really the turning point for my career development. It's really learn a lot of molecular genetic tools, including using viral vectors. And after I moved to UCI, we still collaborated quite a bit, and so as my lab also contributed to the improved the monosynaptic rabies system as the paper published in Cell Reports. And I do want to say a little bit, talk a little bit about the genetic modified rabies, because so far genetic modified rabies is considered the best for the neurocircuit study in the field. The genetic, so the rabies, genetic rabies, it was earlier was German group was modified. They actually cloned they made a clone of the rabies, and they also make the GD-elite rabies, put the GAP, put the GAP, all the things. But we at the Caraway's lab was the first lab to apply the GD-elite rabies for neurocirculation. So I just want to see here is actually rabies. So rabies is related to a simple virus. So we have five genes, and the glycoprotein gene is essential. Glycoprotein allow the rabies to get into the neuronal cells. Without the glycoprotein cells, those rabies will not be able to infect the neurons. And so in this case, glycoprotein coatings also very important to allow, otherwise the rabies virus will not be able to bud out, will not be propagate. And so this is, as you can, and it's nice thing that even without glycoproteins, the RNA, the rabies RNA genome, they will be able to replicate themselves. So rabies virus is really nice self-amplifying markers. So in this case, injection, the registering that time injected the rabies virus into LGN, label bunch, Celamus, label a bunch of the retrograde projecting the cortical neurons. Looks beautiful. Label a lot of these cell body, dendrites, and axons, and you can zoom in. So essentially, like you can see those very much details, like fluorescent version of Golgi stain. But this is much better than Golgi stain. Golgi stain is just random. We don't, you cannot control. In this case, we know those, what type of neurons. They're clear. There's a retrograde labeled projection neurons. And so this is it just starts. And the real thing is that they take advantage of the ability targeting rabies infection to specific cell type, use N-way serotype, and to limit transsynaptic spread to direct input by using glycoprotein gene-deleted rabies virus and transcommutation. So basically, these are two clever genetic tricks. First, what they do is basically serotypes of rabies. So this is wild type. This is you have GDDC rabies. You can grow them in wild type envelope or use the envy. Envy is basically there's a viral coat from the bird, normally from bird virus. So mammalian neurons do not have this bird virus receptor. So if you inject this virus into wild type, brains, mammalian brain, it will not affect. So this is first thing. This is important. This serotype the rabies allow for the genetic testing because you can express. You can use genetic ways. Either in slice, you can do gene gun. In vivo, you can use herpes, transdermice. So allow those cells, specific cell type, to express TVA. TVA is a receptor for envy, so those viruses, the rabies, can get in. And because it's a GDDH rabies virus, right? And if without the glycoprotein, it will not bother out. But you add a glycoprotein here, so these rabies will become an infection particle. They can get into the direct connected presynaptic neurons, but they cannot jump anymore because there's no glycoprotein here. So this is very clever, and these and they test out. They took them. I was there to really even what they started this earlier than that, even before that, 2000, and it really was since working start to work right almost just right before I moved to UCI. Right? And so, but it is a great tool, and it's now been well developed, improved, and applied to a lot of neuroscience and neuroscience studies. And so, in next few slides, I'm going to illustrate how we're going to use these tools to map, to identify new brain circuits. So so we had this. Hippocampus is well studied brain regions. Even I show you the hippocampus joints earlier by Carr and Golgi the joint here. And so it's known the hippocampus has canonical circuits, essentially most fit for internal codecs to dendrites and CA3. And CA1 and CA1 can project to significant. Particularly CA1, it's been much believed that CA1 have very strong unidirectional feed for the projection to significant. And the significant considered as output of hippocampal regions. However, the increasing evidence over the last few decades suggests that it may have a non-canonic back projection from this region to CA1. But previous studies, those are vocal tracers, other things, or electrophysiology. It just, those are less strict methods, not very sensitive enough. It just there's no conclusive evidence to support this pathway until we use monocytic rabies tracing. So we use this rabies tracing system to establish the non-canonic significant projections to CA1. So the idea is simple, right? Because if use rabies, we know it's monosynaptic. If we inject the rabies monosynaptic traces, if we see labeled neurons in sputum, that's very simple. Right? We establish the circuits. So let's see how it will work. So my student injected the rabies tracers with herpes and rabies monosynaptic rabies. So those are we call the injection site, and you can see that because we can identify starting neurons, right? So what means starting neurons? These neurons they express rabies. They also have the machinery from the herpes with TVA, glycoprotein and TVA. So yeah, without things in there, if you look at it, there's actually significant number of sputum neurons labeled, right? Different section, different location of the brain forms monosynaptic rabies tracing in CA1. And then so we also validate the anatomical tracing using the slice-based physiological recording. Right? So I will not get into detail because this study was published last year. This actually is a highly publicized publication. And then we'd like to go forward. Right? Well, we know that yes, we established this pathway, right? And we would like to see, well, what about these what about those neuron project? What about, are these a unique type of sleeping neurons? How are we going to do that? So we use combined viral tracings. So we need to add, because we need first, in order to do the rabies tracing, we would like to label those specific type of neurons with some certain tag. In this case, it's labeled with CRE. So we first inject the CAV is adenovirus. Canine is from dog, right? Canine adenovirus is adenovirus. It's been known that it's respiratory transport, relative efficiency with the payload. In this case, with recombinase. So injected here, you actually label those CA1 projection speaking, and then you can apply, the QE-dependent monocyclic tracing. So you see things that work well. If you look at these injection sites, you can see that a handful of the stadiums. It's important because you see the viral tracing, we'll be able to understand how many stadiums, and also quantify the presynaptic label neuron in different brain regions. So we'll be able to quantify, the network connectivity strengths, and we call the connectivity strength index, right? So you'll be able to really, although we just still do anatomic tracing, but we'll be able to come up with quantitative or semi-quantitative ways to quantify the network connections. Right. So this, and we apply. So we tracing out, to map all the different bone region, the input in different bone regions, and then we compare with overall larger population split neurons. So very much similar. We're not going too much. Just showing that what we can do. That now we can calculate, basically quantify the connectivity strengths based on two types of neurons. One is a general type of exciting neurons, the other one specific type of sleeping neurons. And actually see great, difference. Even those neurons located in the same microcircuits, the connectivities with other bone regions are quite different. And then this is just use the way we know those neurons, where they get input, but also very interesting to see where those neurons project. So we need to use the herpes H129 strain to map output projections of these interesting CA1 projecting sleeping neurons. And Dave Anderson's lab has develop a new condition. This is a condition of the basic pre-dependent of H129 expressed C tomato. And then, so this is conditional. You need a CRE, you will get infected express. But once you get CRE converted, it becomes just like wild type, right? So you have a propagate, depend on time, and therefore further time you'll probably, it's polysynaptic. It's not ideal, but what we work out is that we found out 48 hours seems like labeled, a direct postsynaptic once synaptic spread. So in this case, we first label these neurons and we inject the QE-dependent H129, the polysynaptic traces. Right? So yeah, pretty well. So injecting in speculum, you can see those back projection. You see the postsynaptic label neurons in CA1. So it's kind of very much validates that the CA1 projecting speculum. However, I do want to point out one important thing that even look at injection, even after 48 hours, you will see a lot of neurons dying, lytic neurons here. So this is those traces that are significant cytotoxicities. So people already know that they don't just kill neurons. Even most animal injected those, they will not live. They basically die within a few days, no more than 7 days. So this is one of the major issues for herpes-based viral traces. And for these studies, very important thing that we actually found that those C1 projecting, they projecting to the parietal cortex. In the area we know that in neuroscience, strong implicated object placement. So this is very interesting, right? That means because we know that, we also test out use epigenetic to validate these new pathways, functionals. And this is object place learning is very interesting to the Alzheimer's disease research because one of the earlier impairment for Alzheimer's disease is the impairment object location, object place, right? Such as, where are my keys, right? That's not a normal, right? Those are and so right now, so we identify, so far I illustrate is how we use the rabies tracing and herpes viral tracing to understand, to map all the circuits and do the behavior testing, and currently we will try to apply these try to map these things in Alzheimer's disease mouse models, and also try to maneuver pathway to see if we can enhance the learning performance in AD mice by targeting this pathway. And in next, say 5 or 10 minutes, I will quickly go over our current efforts at the UCI developing the new viral tools. So I probably will not go into read this, right? So throughout the talk so far, you want to know that there's successful rabies tracing, very successful for retrograde, very successful, but antegrade is pretty much developed. So one of the direction here is where we are going to develop a new much safer, effective, validated antegrade traces using based on the Hilby and H1 strain. And the second direction is that basically rabies still have rabies is a great choice to work on, but we like to expand, optimize this. And particularly, we like to reduce rabies toxicities for long-term repeated longitudinal imaging and recording in vivo. So the antegrade monosynaptic tracers, H120 tracers, was developed by my colleague, my collaborator, Dr. Mingfang Rong in China. Right. So as you can see, that pretty much this is actually pretty much is motivated by the rabies monosynaptic rabies development. So you basically create conditional deletion mutant and make a conditional replication. But anyhow, so this is really so it's basically so we can create those TK mutant, and then we can add the EJP. So this can get in, but without TK, because usually neuron don't have a very low or minimum TK, they will not replicate unless we put the trans component, put the TK here. Right. So this is essentially like work as monosynaptic rabies instead of another direction for antegrade. So as you can see here, this is injection sites. Right? Overall, it's principal work. Right? It's basically proof of principle single work, but you see still a lot of neuron dying here. Right? So this is not still high efficacy. Of course, it's great progress. We want to make it antegrade monosynaptic spread, right? Because it's genetically controlled monosynaptic spread, not like time, like the other tracer you just roughly based on time. So we good news that we got fund. We have organized a team. We submitted a proposal last year. We got a substantial funding. Right? We got 3.5 million for 3 years to develop second generation antegrade monosynaptic tracer. And this involve 4 groups. And our recent teacher, Dr. Seminars Lab, joined us in this project. And we proposal to test the characterize traces in both rodents and non-human primates. And our goal, we hope that we will be able to accomplish our specific aims, and this new tool will have broad impact, right? Not just to do anatomic tracing, also enable optical imaging, physiological recording, activity manipulation of defined antegrade projection networks. And so, regarding the second directions, so both Dr. Seminars has been instrumental to these directions, and particularly both postdoc Alex has been leading the charge. And we know that the EJP variant very much expanding the color palette. This was the need for us to really use the newly improved fluorescent protein to create different color variants. Right? That's important. Another thing that because the way we studied the rabies, That's a study that we're going to look at high resolution. Very interesting to have different cellular representation, say nuclear localized, membrane localized, and cytoplasm localized, and also interesting to look at presynaptic, postsynaptic, like a PhD 95 fuse and synaptophysin fold. And also we're interested to look into the labeling specific cellular organelles, such as mitochondria and other things. So this will be great, not just for health sequencing studies, and you can see the UCI is very strong in aging and Alzheimer's disease. Those two will become very well to use for studies of disease brain structures. And in addition, so far a lot of the rabies tracing mostly for enotopic tracing for fluorescent labeling, right? So basically used for light scope. Light is so optical imaging use, and when in this case we can generate those ferroparameters. Right? Ferritin, we know ferritins can be used for EM study and also for the in vitro, in vivo magnetic resonance imaging. And so yeah, as shown up here, we actually have this. We made a very good progress. Those done most of these, and this is very exciting. Right, this is done at the UC. We inject animals, send a sample to UCSD. You can see those. You can see labeling exon, and particularly exon terminals. A lot of ferritin conjugate. And so why is this relatively preliminary, but this clearly this is very promising. Right? And I will not get out here, just say that the rabies, people is one thing that with how to reduce rabies toxicities, right? So far, all the paper published, they still we could not replicate, and the field still could not replicate. So we are taking new strategies, right? And instead of people, normally people take a look at the L or other protein. Most people look at it, and so, but Dr. Samuel's lab is, expert in the MI virus, modification, right? So we use we use their expertise to control the toxicities, and we're focusing on the P protein, right? So for RB4, so we can either use the kind of iris constructor, or we can also introduce parallels. We can do the site-directed mutagenesis. And so we're not, so good thing that we're not the disease is, alone. We actually have our center, the newly established centers, provide a very strong infrastructure for us to do to perform the proposed work. And particularly, we have our own viral production facilities at the Center for Neuroscience Mapping. Right. And yes, these centers, we have the production facility, and so it's very important for distributing our viral vectors to the neuroscience communities per the grant requirement, but also the center facility will be able to support the UCI's basic and clinical investigators use viral genetic tools. And if you're interested in that, just go to the website. You can download the form. Just fill, it's very simple. Just fill, if you need, you don't, just fill out this and let us, staff here will get back to you. And so you can see it's very simple. We can actually, so far we actually been take quite, I would say, a significant number of orders. Yeah. I will not go anyhow. So yes, with a greater number of the funding project, we do want, we have more. We like to have invite, more people. Invite molecular biologists, virologists to join us, to pursue this very exciting project. And finally, I would like to thank the people in my lab. So as you can see, my lab, we have a relatively young team, but my postdoc, graduate student, technician, they work very, work, not just work hard, work smart, work effective to get things done, very well. Yeah. And then you also can see that we have I also contribute the success so far to a large number of wonderful colleagues. Right? Without them, I think things will not be so far and also so interesting. Thank you both. I think if maybe, I know it's a little bit long, but anyhow, I'll be happy to take your questions.
Thank you very much, Xiangmin. We do have some questions. If you look on your chat box, there's one from Yongsheng Shi.
Yeah.
It's in Chinese characters, so I can't ask that.
Yeah. So, Yongxin, why don't you unmute yourself?
Yeah. So, sorry, I was saying hi to another audience member. It was just not meant to send to everyone. Sorry.
Okay. Yeah.
Yeah.
Yongxin means 认真学, so means learn something carefully. Do you mean right? Yeah. So.
Yeah. I'm here to learn. So that's what I'm saying. While I'm here, so I want to ask a question. So when you inject a herpes virus into these brains or neurons, do you get dormancy or latency, or is it just a high amount so that you don't get that?
Yeah. So usually this happens with the peripheral, right? Because we have a herpes experts in was in right. So use peripheral, you have dominant states production productive infections, and because we directly inject the brain, right, to infect neurons. So mostly you don't it's we actually worry about the infection is so strong, right, in kill neurons. So it's not a opposite issue for us. We like to have control the expression, the replication, control replication to maintain cell health. Yeah.
And Paul Gershon has a question. Paul, do you want to come on and ask that question about the time resolution?
Yes, can you hear me?
Yes.
So I guess the ideal situation would be able to eventually have a virus that can jump transsynaptically and then map in real time, right? But I think you kind of answered the question of the future there by mentioning functional magnetic resonance imaging. Is that the direction this is going in? That
so
you really watch the wave of virus spread out in real time over a more complex network?
Right. So Paul, this is a great question. So, but the first, I will take you answer your question in two parts. First part, what we want to establish monosynaptic, strict monosynaptic through genetic control. Right. This is very important because with polysynapse jumping, you just don't know. After 1 or 2, you don't know where those connections come from.
It increases exponentially, right?
Right.
So
monosynaptic, strict monosynaptic genetic control. But rabies is doing pretty well, but still has certain issue, peripheral steps issue. And for herpes, for the H129, now Mingfang's lab make a great breakthrough. We know that some of the wild type of H129 do have potential delay the retrograde, although it's preferential retrograde. But so now we found a certain gene called GK actually can pretty much remove the retrograde potential. And also now we have new version based on the GK, we can make a really nice antegrade monosynaptic. Less toxicity, but much more efficient label. That's the thing. And you mentioned this actually is a great idea. What we can do is that when I'm thinking about it, some way use the inducible methods. After you pass one and then add another jumper, that will be basically
nice. Yeah.
That's why I think is I told in the beginning, I said, well, I would like to take this opportunity to interact and collaborate with more viral experts on campus, right, including yourself.
And just out of interest, what is the time resolution of MRI? I mean, if you wanted to look at the rate of transmission along different types of axons, would you be able to do that?
So right now, I'm preferred because herpes kill, if you use white type jumper, very much you get once you get a brainstem, you kill animal, right? So no more than a few days. Right? And rabies okay, but rabies also the same thing. You propagate to animal to brain, particularly injectable brain.
Yeah.
Right? So what we're doing MRI is that in order to allow us for MRI applications, we put the ferritin.
Yeah.
So and the rabies nice that the G delete rabies, they replicate in the cell. They generate sufficient enough ferritin allowed MRI detection. Previous vector people try will not work because of that. And so and what we do that we emphasize we can do repeat longitudinal within animal imaging. That's the point. That's where something you say, well, how that developmental aging disease AD. So that's why both Bert and I are very excited, and we hope we'll be able to accomplish our specific aims.
It's fascinating. Thank you. Yeah.
Xiangmin, we've got a question from Hugh Davies. He wants to know, has pseudorabies virus been used as a vector for vaccine antigen? And is it inflammatory?
Interesting. Here I think it must be the pseudorabies. One thing that I mentioned, that pseudorabies does not infect primates, right? So that's the downside, right? So even like Basa strain used successful for rodents, but if people use the non-human primates, it will not work. So I'm actually not an expert in this area. So I think you're right. You may do Google it, you may talk to other people, or maybe other people can and
Roz has a comment.
Roz, okay.
I have a comment. The Bartha strain that of pseudorabies that Xiangmin started with is the vaccine strain that is used in pigs. So it is already a vaccine.
So it must not be very inflammatory either.
Well, that depends. Yes and no. It depends on the dose.
Yeah, they basically use for pig, right? Because pseudorabies they usually.
Yeah, it's a swine virus.
Yeah, it's not for. I guess human. You are talking about the human, am I right?
Yeah, I was just curious.
Yeah, humans.
I mean,
general people believe pseudorabies does not infect primates.
okay.
And I have a question, Xiangmin. Hugh, you have something else?
No, I was just thinking, if it did, it would be a useful vector for targeting things like neuroblastomas and, Alzheimer's maybe, I don't know.
Hugh, can you speak up? I cannot hear you.
Oh, can you hear me now?
No, I can. Why don't you repeat your question, please?
Let's see, hang on. Is that any better?
Yeah, we can hear you, Hugh.
Oh, okay. So I was just curious if it would be a useful vector. If it doesn't infect human cells, it's probably not going to be of any use, but maybe in animal models. I was thinking, there are plenty of tumors in the nervous system that could be targeted using a vector like this.
Yeah, I think it might be interesting. Yeah.
Anyway, I had just a question. I should probably know the answer to this, but I'm sure you do. What's the basis of anterior grade versus retrograde transmission of these different viruses?
That's $1 million question, and I think really a big prize, right? So that's
that's not known.
For certain things we know. Basa strain we know because as US the link accuracy work showing that the US genes really is critical, right? So it's related with the axonal antegrade retrograde molecular transport, right? We know rabies people pretty much believe specific because of the rabies binding to the molecular motors in axonal transport. But for the herpes, it's very interesting. Roz can chime in. Very interesting. Herpes, you can go retrograde and antegrade, and depend on the activation states. If we can figure out these, we can apply to neuroscience or even for others. There's much more beyond the neuroscience. Yeah. So those are not just virus. This is really fundamental mechanism that we can apply for many things.
I will just chime in and tell Xiangmin, because I don't think he realizes this, but we just hired as an assistant professor a postdoc from Lynn Enquist's lab, who is going to join us in January. Her name is Orkida Koyansu, and she will continue the studies looking at transmission in these neuronal cultures that she uses with both herpes simplex virus and pseudorabies.
That would be great addition. Yeah.
We'll introduce her to you through Zoom when she arrives.
Yeah, definitely we should talk, maybe in December. We have not before, we should talk. Yeah. Thank you.
Anybody else have questions for Xiangmin? Well, if not, then I want to say thank you very much for really a terrific seminar and very informative. And we look forward to your progress. And I, of course, look forward to collaborating with you in the future.
Thank you, everyone.
All right.
See you next time. Bye-bye.